Howdy, this is Dr. Aguilar's lab at Texas A&M University working with ATP-Bio. In this video, we're going to go over thermometry using laser beam splitting techniques to improve rapid and uniform warming. In this experiment, we aim to observe the enhanced laser warming of deionized water with the assistance of titanium nitride plasmatic nanoparticles and a multi-laser spot optical system in order to achieve a uniform thermal distribution along the nanoparticle water solution. The ultimate goal of this experiment is to be able to rewarm cryopreserved microorganisms using these same conditions to achieve high viability of small specimens such as fish embryos, mosquito larvae, or artemia after the laser rewarming process. Titanium nitride nanoparticles are provided by Lorenzo Mangolini's lab located at the University of California, Riverside. These nanomaterials are synthesized with the non-thermal plasma process, which leads to the formation of freestanding titanium nitride particles with crystalline structures that are below 50 nanometers in size. Extinction measurements of the as-synthesized particles show a clear plasmonic resonance in the near-infrared region with a peak plasma position at 800 nanometers. Our first step in this experiment is to prepare the titanium nitride nanoparticle solutions for the laser warming experiments. Using an analytical balance scale, we weighed 0.001 grams of titanium nitride nanoparticles on an anti-static boat. Then we placed the nanoparticles in 5 milliliters of deionized water to make a 200 microgram per milliliter stock solution in a glass vial. The vial was sonicated for 5 minutes to obtain a homogeneous solution. Sometimes some particles did not dissolve fully, so we used a vortexer for another minute to help dissolve the remaining particles. From the stock solution, we diluted to obtain 3 solutions with concentrations of 12.5, 25, and 50 micrograms per milliliter. One more time, we used the vortexer to assure a homogeneous solution, and now we have our samples ready for the laser warming experiment. Our optical setup consists of a continuous wave laser with a wavelength of 808 nanometers. This is similar to the resonance plasmon peak position of the titanium nitride nanoparticles. We also had a fiber collimator, a lens with a focal length of 100 millimeters, as well as 3 50-50 beam splitters and 4 mirrors, which were used to help equally divide and direct the energy laser into 4 identical spots. A quartz cuvette is then placed at the center of the 4 laser spots paths. The beam splitters divide the laser beam in 2 different directions. The mirrors were used to reflect the laser and direct it towards 2 sides of the cuvette in the center. 2 of the beam splitters also directly directed the beam into the 2 remaining sides of the cuvette, allowing the beam to hit all 4 sides of the cuvette at once. Below the cuvette, we placed a small magnetic stirrer, and inside the solution, a magnetic rod to avoid material precipitation. One of the dilutions of the nanoparticle water solutions was then placed into the cuvette. To measure the average temperature of the solution, we used a set of 4 thermocouples that were submerged in the solution and were equally distanced. The thermocouples are connected to a temperature reader to see the incremental increases of the temperature in time as the solution is heated up by the laser. The thermocouples are mounted on a micrometer stage to help carefully displace them to different positions along the cuvette, and like this, obtain the temperature rate of the solution along the whole volume. This laser warming process was allowed to run for 10 minutes. Here you can see the initial temperature recorded by the thermocouples, which recorded temperatures for the entire duration of the 10-minute process. After 10 minutes, the laser system was turned off, and the thermocouple readings were stopped to analyze the data. As you can see here, there was a small incremental increase in that 10-minute duration. After analyzing the data, we were able to determine that the heating rate increases when you use 4 laser beams compared to 1. Overall, we follow the thermometry and laser warming techniques presented in this video to study titanium nitride nanomaterials as an alternative material that can be used to rewarm vitrified biological samples. As you can see in this figure, titanium nitride nanoparticles and clusters presented higher heating rates than gold nanorods. These techniques can be used by other researchers that would like to study photothermal properties of nanoparticles. Rewarming of small biological systems in the droplet scale remains a challenge as vitrification at low CPA concentrations is possible. The rate and uniformity of rewarming from the cryogenic state affects the viability, physical, and functionality of the biological system. Therefore, this laser setup is an initial step in designing a warming technique that can be used to nano-warm droplet-scale systems after vitrification to improve the speed and uniformity of heating with the 4 beams that can result in higher viability. Additionally, this was used to predetermine the potential for titanium nitride to be used as an alternative material for laser nano-warming. These results highlight a promising alternative plasmonic nanomaterial to rewarm cryopreserved biological systems in the future. As seen in the droplets here, titanium nitride showed successful vitrification and laser warming without ice formation. These techniques can be used to improve the viability of biological systems such as artemia and other post-vitrification and warming in the future. Thank you.